comparing to Lisa and Eugene, I think I'm going back to eukaryotic domain a little bit. This is work actually is done with my colleague from water survey at the Illinois, and also with David, and also I forgot your name, just joking. Nico is at JGI. And the reason is Nico actually they published a paper in Nature, and actually they reported the biggest one of the biggest virus contiguous genome 500 plus. Actually, they came out from the reactors in my lab. It's running on that. And this became a real Nature collaborations after that. All right. So if you've been to Chicago, actually, you know, actually, this is you're actually looking from Navy Pier into the skyline of Chicago. Okay. And I'm trying to see how we can actually use the viral profile to start to indicate the water quality actually going on in the waterway. Okay, big green. This is hot summer day in the end. And you see a bunch of crazy or maybe exciting people jump into the ship canal. Actually, a bunch of them are politicians, Chicago politicians. No wonder. But you know, there is reason actually, and it has to go back to history actually, reason for that. It was before 1900, and you can see the Chicago River. Oh, sorry, let me go back. Okay. This is Chicago River. This is Calif River actually, and actually at the time actually people just discharge all the sewer actually into the river, and they flow into Michigan, Lake Michigan, and that's why there's outbreak of cholera, and actually many people actually die maybe because of enteric viruses as well. So what did they decide to do? They decided to do it this way. There's a really marvelous civil events going on. They create they create the canal, and we call it the sanitation ship canal, connect the Chicago River all the way to Illinois. They also bring the Calumet through this Calif canal and combine and go in there as well. Okay, so then what has happened? So all the sewer is now flowing to Lake Michigan, actually flowing to St. Louis, Missouri. And you think they are okay? Of course, not okay. Even until now, actually, so what happens at the Gulf of Mexico, then they are now okay as well, because the hypoxia actually is a real issue, and major contribution actually is from here. So what has happened is actually, so there are actually major outflow for wastewater, which is treated wastewater actually discharge into these ship canals, and there's 1, 2, 3. This is the 3 major one. The first one actually is the largest wastewater in the entire world. Okay. So this is how they look like. This is Lake Michigan, and this actually is where those people jump into it. This is Calif, and actually at the end, actually this ship canal, this is Lockport Dam, and everything is just discharged into the Illinois River. This is where you can regulate the barge up and down to the Lake Michigan. And actually this outlook from State Park into the Illinois rivers here. What we did is actually, if you look at the volume actually, so this is probably less than 1% of volume of the water is coming from Lake Michigan here. And then after the first discharge, the volume increased, the second discharge increased to 80 plus, and third discharge, and it's 100%, almost 100% wastewater discharge until this Lockport Dam, and the volume started increasing into the Illinois River and also Mississippi as well. So what we did actually, so people want to find out how good the quality is, and then usually what you do, you actually use the chemical as well as physical and some microbial like indicators like fecal and total coliforms. The question is are they sufficient? Are there alternative criteria we can use? And people are trying to use bacterial populations or indicators and using sex-sense biomarker. Maybe we think actually viral profile can be another good biomarkers for that. So what we've done actually, we took samples along this ship canal actually from the Lake Michigan, and then the second one upstream of the major wastewater outflow, and the downstream, and from Calif and Merge, and before they go out to the Lac Borton, and then the Illinois River here, and also pristine, it's inside the state park, so it's a pristine water actually. And also downstream of Illinois River at Havana, and also Mississippi as well. Okay, and we sample at 2 different events, it's 2014 and 2015 for that. So this is sometimes just kind of goes through quickly. So after sampling, sorry for that. We actually separated by bacterial biomass from the virus, and then concentrated and sequenced it, and after that it's all done by David here at JGI. The calling for the contigs also is based on the methods he has developed. And we have found about 37,000 contigs here, greater than 5 kb. And then actually later on we use CRISPR and also another clustering analysis to identify which environment they are associated to as well. Okay, so let's see how it look like. So this again, this is where they are taking along the entire ship canal, and this is from the Lake Michigan all the way to see the water flow. And this is another branch coming in here. And so we will ask actually what are the unique contigs you can find from each sampling site. And that's the bigger the dots is, suggests the more unique contigs from each environment. And the answer is Lake Michigan, as well as the state park and Lake Mississippi. Actually, there are more unique contigs, but not along the ship canal here. It's just very few. So we then ask actually, so how many of that contigs are shared among any 2 given sampling sites? And so we actually take those from the ship canal sampling site and compare with those reference from Lake Michigan, Star Rock, and Havana, and Mississippi, and you find very, very few share in compared to what's shared among those wastewater sampling sites along those ship canal. So this is amazing, actually. So next, what we did is you can, if you have used IMG VR, they actually can classify your contig most likely to a specific environment or habitat. And this is classified based on engineer, aquatic, and terrestrial, and host associated. They can further classify to the next level. And what you see here is all those black bar are the known one, and those white are not unknown. So we can take those known one actually and further classify to which environment or habitat they are. So what you see here is this from here to here, this is all associated with sanitation ship canal, and there's primarily dominated by engineer and wastewater associated, okay? And then some actually with aquatic environments here. And it's definitely, most of them are associated in the Lake Michigan sample is associated with aquatic. All right, so we can look at abundance, and you can see the abundance even further increased for those samples along the ship canals here. So what we did is actually we further cluster all those samples, and they actually show up in 2 big clusters. And you can look at that either as water, wastewater associated, or clean water associated. So what we decided to do is take those those contigs which are from these clusters here with greater than 50 times coverage, and those from here with 50 times coverage. And then we find out the average abundance from this cluster and also their corresponding abundance from this cluster. And this is what we found here. You see two huge clusters here. They're all greater than 50 times coverage here. And use t-test, you actually can find out which contigs are significant. They only more prevalence in here than here, and we can find about 1,000 plus. And this actually indicates how many times coverage of them actually varies from 50 times up to more than 500 times, or some of them actually is 10,000 times actually. So next, what we do is we think this is our wastewater associated virus. So we went back to David's methods. So we did it. So we used the there are his methods, and you can call out those contigs which are based on spatial cluster and say they are truly wastewater associated. But we can actually further define remaining which were not called out. Actually, they're actually also wastewater associated using this method. And maybe this is it can be applied very locally or regional. Whether it's globally or not, I don't know actually. So there are about 700 plus, and then we later on put it back to this one, and you can see those blue bars here increase even more based on counts and based on abundance actually. So this is amazing actually. You can have more than 50% of the content associated with wastewater now based on abundance. So kind of wrap it up actually. Now we understand why people jump into there, and there are also reasons for that. So because they are they are trying to implement actually a treatment efficiency. So this is what I do. They have long-term monitoring program going on, and this is the coliform counts for the last 3 years or 4 years. And what you see here is the average coliform counts is huge actually in 2013, 14. And in this particular place, actually, they start to introduce the disinfections at this wastewater treatment plant. And you can see the count drop to this red line, which is the guideline provided by EPA, saying this is safe for recreational use, actually. But then if you look at the few dots, a few miles down the road, actually, the counts goes up. All right. And this is just where they jump into somewhere here, actually. So anyway, so I think this is just, we haven't completed analysis yet, but I think there are other methods you can look at. Boron is just really good indicators. We use detergent. Actually, it's one of the substance you can find primarily in the detergent. And we found actually the concentration we found in the 2 sampling events, they highly correlated with how much the wastewater are present in the particular given site. And looking at the virulence here, actually, we can tell how good the quality is right away by looking at those unique ones associated with wastewater as the habitats. So, in summary, we think it's just a great way to look at now, actually, besides just looking at evolution and other perspectives. You actually can start to use them as key biomarkers. You can monitor the water quality, maybe quickly using a qPCR if you have a unique virus you would like to detect. We are actually trying to wrap up the work by identifying the host of the dominant virus clusters. And we're trying to link the viral profile to the 16S profile we have analyzed using the co-occurrence network analysis and trying to validate actually what the prediction came out from David using his methods as well. And that's it. Thanks for suggestions. Thank you.